It does all seem very regimented based on that rubric, but the fact of the matter is a lot of times in science there is that degree of subjectivity, where certain rules or certain papers or case studies don't necessarily fall into that perfect little box. And because of that, we do see a lot of, I shouldn't say a lot, but a good amount of subjectivity in our industry. Some of the key factors that actually cause this discrepancy between labs are some issues where, like I had mentioned, the ACMG guidelines are now over 5 years old. So a lot of labs that have been using those ACMG guidelines have kind of modified them individually. And they're not necessarily well published how they are modifying those guidelines. That could very well swing the interpretation of a variant from non-pathogenic to pathogenic of some sort. For example, a lab might treat a patient who has a mild phenotype as a decreased level of evidence, whereas another lab might treat that same patient with a mild phenotype as a higher level of evidence. So the difference in the confidence of that particular patient will cause a variant to either be curated as pathogenic, or it might even drop it all the way down to a variant of uncertain significance. So that's one situation. And like I mentioned before, there are those kind of third-party groups that are working on kind of modifying the ACMG guidelines, which I think is going to be a lot of what the new ACMG guidelines are going to adapt. So that's kind of helped decrease the discordance.